Hello, my little tiddlywinks, and welcome to the first ever episode of a new segment called Science Sunday. It's where I'm going to go ahead and show you some cool science stuff. We'll talk about some sciencey topics, and I can share my love for science with you. Because I am a scientist, believe it or not. I mean, I guess. Mad scientist, perhaps. We'll stick with that for now. So every week what I want to do is on a Sunday, I'll go ahead and make a little presentation for you guys, and we'll talk about different science topics. These topics can include anything that you guys desire. If you want it to be biology, we'll talk about biology things. If you want chemistry, it'll be chemistry. If it's physics, we'll talk about physics. I'm not much of a physicist. I'm not much of a, chemist, but biology is definitely the strong point. But I'll try my best on anything you guys can think of. Any topics you see in the news, if you don't know about maybe Zika, if you don't understand HIV, if you don't understand antibiotics and what's going on in the world with them, put that down in the comments and I will try and appease you guys. I will try and make it so that what you guys are interested in, that's what I will try and do as much as I can. I mean, if you're like, what's a black hole? Well, I'm gonna be a little foggy on that one, not gonna lie. But I will always try my best, and if, you guys aren't coming up with good topics, I'll have to come up with my own. So please, like and comment this video, add some ideas in there if there's anything you guys are interested in, and we'll go from there, alright? So for our first installment of Science Sundays, we're going to go ahead and talk about green fluorescent protein. It's one of the coolest things ever. Okay, it just kind of glows. That's really all it really truly does. But it has a lot of uses. This is the structure of it, and that's kind of boring. It looks kind of neat. I guess, whatever. But what is it? So, some of my biotech peoples out there should already know this. It's one of the biggest deals. You can search 1,000,000,001 papers. Literally, it's like 1,000,000,000 papers that are using green fluorescent proteins right now. It's such a big deal. And what it is they went and discovered in this cute little jellyfish. It's called the crystal jellyfish. They live off the, I believe it's the west coast of North America. Normally they just kind of look like this, but when you put them under ultraviolet light, they glow green, like they're radioactive monsters. It's so cool. The group that discovered them was Isamu. I believe that's how you say his name. I'm not going to pretend not to really know how to speak other languages. But back in 2008, they won the Nobel Prize, which is kind of a really big deal in science. Him and his group, I believe, discovered this species and then went through and kind of discovered everything they really could about green fluorescent protein, or as I'll probably start referring to it as GFP, just to save some time. So, these guys went through this whole amazing process, and they discovered, that cool jellyfish's ability to glow, which is really, really neat. The process that they work with is it uses this aquarin, I believe is how it's pronounced, to interact with Ca2+ ions. And this creates a little bit of blue light. It's not a lot, but it's enough energy that can then transfer over to GFP and create that green glow that we saw back here, which is super cool. But the downfall of it, in its wild type form, in that jellyfish, is you do create two peaks when you're looking at the intensity of the light. So you get this blue peak here and a green peak there. And when you're trying to look at things doing spectral analysis, this can be a bit of a pain in the butt, because you don't want to have two colors, especially if you want to use multiple colors to look at later on. So we've gone and we've created our own GFP, because we're a bunch of mad scientists who do what we want. But we made more colors. So we extracted colors from different organisms. So from a lot of the jellyfish family, you get a lot of these blues and green kind of colors, and even some of the, M banana. I don't quite know why, but we decided to name all of the colors after fruits, which is, I guess, interesting. But yeah, so you've got your honeydew, your banana, your tomato. Yes, a tomato is a fruit. It is not a vegetable. Just laying that out there, but you should never put a tomato in a fruit salad. That's a horrible idea. Anywho, so yeah, they got these guys from, like, jellyfish. They also extracted a lot of them from coral. They found that a lot of the corals have red and orange fluorescence, which is super neat, and you can actually see that. You could use ultraviolet light on some corals, and they'll actually glow in the dark, which is super neato. You can see, like, these are actually glowing. These aren't just the color of what they look like. Generally, they don't have much color, but once you put them under that fluorescent light, that UV light, your black light at home, they will glow these cool colors. Now, we use them for all sorts of things. One of the most basic that we've done is we've put them into plasmids. For those of you that don't know what a plasmid is, it's what bacteria have. I mean, they have their normal DNA, but they also have plasmids. It's an extra piece of DNA that's not necessary for them to survive, but it's a circular piece of DNA, so it's not like ours. Ours is like this guy here, where it's just kind of linear, the ends don't connect, but with plasmids they do, which is super neato. But what we've done is we've added the GFP gene into plasmids as a selective agent. So, like in this case, this is the P-glow plasmid. It's one of the most commonly used ones now. It's got an ampicillin resistance gene. So ampicillin is a type of antibiotic that we use to inhibit other bacteria from growing on the plates that we're using. So just the ones with our plasmid will grow. But we've also added in that GFP gene over here. So what happens is, if the cell has this particular gene, it will glow green. So we can actually select colonies that have this plasmid, which is super useful, because sometimes we might want to add in a gene somewhere of interest. So maybe a gene for, I don't know, cancer. A magical cancer gene. If we want to see it, that we can put it into a bacteria, we can splice it in at one of these special locations called restriction sites, like this NDE1. We can cut the DNA here and throw in a gene, and then we can select for it using this GFP, because it's really easy to tell if the cell picked up this plasmid because it glows. And it's pretty cool. So you can see here's a whole bunch of bacteria. This is just a really great image. I forget which lab has actually made that one, but it's a really famous one. But you can see they're using blue fluorescent protein, a dark green, a light green, they've got a red, they've got purples, they've got browns, they did all sorts of colors. Another lab went and did a really cool Christmas one, where they did a bunch of these really nice ones with red and green fluorescent. And this is bacteria. These are literally just whole bunches of bacterial cells clumped together that they just literally painted onto plates. Now this image down here was for, I believe it's Albert Einstein's 150th anniversary or birthday or whatever. They actually went and made these images. Basically, it's just a bunch of dots of bacteria. So they use just these guys. So there's a red and there's a green. And they went through and they actually created an image of Albert Einstein out of bacteria using fluorescent protein. So this is just artwork. It's nothing crazy. We didn't do anything cool. These were just to have fun. But we do them for all sorts of very, like I said, selective agents. We can pick out particular colonies. But we've done a lot more. So this is fluorescent microscopy. It's the method of using UV light to cause certain proteins to express color under the microscope. So you have to use a really fancy microscope to do it. But you get these amazingly beautiful images. I love these to death. These are one of my favoriteest things ever. And what you do is you can take something like GFP and attach it to an antibody, add it to your sample, those antibodies will be attached to the very particular things that they're interested in. Like in this case, we have GFP, I believe it was GFP, is attaching to the antibody, which attaches to the cytoskeleton. So you can see here all these green filaments. That's the cytoskeleton. These are each of these clumps here. Each of those is a cell. And these green fibers are that cytoskeleton, structure that holds the cell shape. Now they all also gone in and put in a red fluorescence into the mitochondria. So each of these little red dots in each of these cells, those are mitochondria. And they've also done a blue fluorescence into, I believe in this case, it would be the DNA. So it's actually staining this nucleus blue. So this is really nice. I mean, it's good to be able to look at cells, but they're not that interesting, and it's hard. You can't really see much of the structure inside. But with this, you can see absolutely amazing detail and super vivid colors, and it's beautiful. And I suggest, if you want this kind of image, just go search fluorescent microscopy somewhere, you know, Google Images or whatnot. They're absolutely beautiful, the colors, and you can use so many different colors on one cell. It's so neat. But we've done other stuff. So this is a fruit fly, or a fruit fly head at least. You can see here, here's the main head part right in the middle, and then each of these is an eye. And what they did is they went into the embryo, when it was first developing, in the area they knew would be the head, and they added in a fluorescent gene. So one of them was red, one of them was yellow, one of them was blue, to different cells at the very early steps. And then they let that fruit fly develop into an adult, killed it off, and then took this image of its brain. Now somewhere, probably like right here, that was where the first red cell started. That first cell they added red fluorescent protein to. And it allowed it to grow, and that one in particular turned into the eye. So the eye nerves in particular. And this yellow one that they started out with turned into this eye, and a blue one turned into certain nerves there, and this green one went here, and this yellow one went here, and light blue went there. And they did all these different colors to see what cell turns into what. So that hopefully they can use that information in developmental bio to see ways to help, you know, possible future children or organisms that could have birth defects. And we can see where, you know, oh, this one cell, if that one gets damaged, it leads to damage in the eye. If this one particular cell gets damaged, it leads to this kind of nerve damage, or this or that. So it's really cool. And there's loads of rainbows. So this one just happens to be the fruit fly. It's a really cool image. This is a zebrafish. So here's an eye, there's the spine. You can see all the different nerves in the brain and down the spine. Absolutely cool. They've done some in human development. Super amazing images of the brains. So cool. Very worth looking into. So those are brain bows. We've also done some other really cool developmental stuff. Not too long ago, somebody discovered a particular type of GFP. It's a scleraxis GFP. And it only binds to nerve cells. So this is a mouse leg, a mouse embryo, so it's still pretty young. But what they've done is they've got one to stain these muscles red, so it's a red fluorescent protein. So each of these is muscle. They got the tendons in green, so you can see like down here, all these are tendons. And the nerves were stained blue. So they can actually watch the development of this mouse's leg over time and take pictures. Usually I believe they do have to kill the mouse to get it to do that. Not super fun, you know, but they're just mice. It's okay, I guess. You know, I don't like killing animals either, but it's kind of a necessary evil for science. And, you know, if you guys want to discuss animal use in science, please leave a comment. Let me know, and I can try and shed a little light on my views on it. I'll listen to your guys' comments on it, and we'll see what, you know, the world thinks. But this is really neat. We can actually watch the development of this entire leg, watch all the muscles, watch the nerves growing. And that can give us some pretty big insight on hopefully helping us be able to help cure some problems that do occur. Some other really neat things. This is really fancy stuff that I know nothing about. But this is an electron microscope image. So I believe this right here would be the nucleus. We have the endoplasmic reticulum and stuff like that sticking out here. But you don't get great resolution, and it's really hard sometimes to really tell which parts are which. What they went and did is they added fluorescent proteins with antibodies to go and attach to different structures that they knew would go there. You know, we have antibodies for pretty much everything. It's quite impressive. But what they did is that each structure got a different color. And they used a fancy electron tomography to take a picture. And these are supposed to be roughly the same thing. Couldn't find an exact picture. But you can see that they actually got more of a 3D image. And you can physically tell, like, this part here is different than this part, is different than this part, is different than that part. Which is super duper neat, because now you can actually distinguish more particulars than just, this is part of a cell. No, now we can go, oh, well, this is this very particular part, this is this very particular part, which is really neat, and I love it. Cancer research has really used GFP. It's probably the biggest user of GFP because it's so useful. It really is. And like I said, there are a bajillion and one papers out there that use GFP and other fluorescent proteins to help with their research. So in this little guy up here, you can see each of these colors, those are a tumor. So they actually injected them right under the skin, and they can actually watch where these go. So if a piece of this were to break off and metastasize and go elsewhere, they can actually see that little green dot. Like up here, this little guy has brain cancer. I don't think these are the same mice, but it was from the same research. So they actually had a piece of this metastasize and left and actually reattached somewhere in the brain. And they can actually see that with just light, using that ultraviolet and really fancy cameras. But they've done other things. So one of the biggest issues with cancer is they do this process called angiogenesis. It's where they start recruiting new blood vessels. They cause new blood systems to grow around that tumor to feed it. If you can find a way to stop the angiogenesis, the tumor won't progress because it can't get any bigger because it's not getting enough food. So one thing they did was they went and stained, or they added red fluorescence to all the cancer cells. But then to all veins and arteries and stuff like that, they had green fluorescent, which turns kind of yellow in this case. But each of these is an actual blood vessel, or blood system, or whatever you want to call it. But they can actually physically watch the growth and the pattern of how these grow, in hopes that we can find a way of preventing new blood from getting to the tumor, so the tumors don't grow any more than they already are. Because if they stay small, it's not really a big deal. It's only when they start getting too big that they cause all the big problems. So this is a really cool way that they're starting to use that fluorescent protein. Now we have transgenic pets and animals. It's where you can add DNA from something else into something. In this case, they've done it in a whole bunch of stuff. We've done it in mice. You can see here, you can see in this picture, it's kind of a double imposed. This is what you usually normally see. So they usually use like naked mice. They don't have any fur. That actually occurs normally, apparently, out in nature. There actually is organisms that don't have fur. It's kind of creepy, like naked mole rats. It's gross, they're weird looking. But you can put in the fluorescent protein, and you can express it under the skin. It won't express in the fur. That's one of the big things, and I'm not quite sure how they really did this one with the rabbit. I believe her name's Alba or something like that. I don't really know how they did that one. But normally, they're hairless, and you can see the color come through under the black light. You have like, these kittens here, you can see that their nose and their ears are really what's glowing red. Or this little girl here, Ruby. She's a sweet little beagle that they added red fluorescence to. And like you can see here, in the day, you can't see any real color. But once you put them under the black light, things like her toes, and her feet pads, and her nose and stuff, they will actually glow red under UV. Which is cool. You know, there's no real big use for it yet, other than it looks neat. But later on, they will have really cool things. So, like these guys here, you can see, they just glow and it's fun. Nothing neat, but you can't get any of these in the United States yet. They're so far banned. I believe over in like China and some of the Asian areas, you can get some of these glow-in-the-dark mice, I believe, and some of the rabbits, and I think like guinea pigs and stuff. But not in the United States. What you can get in the United States, though, are glowfish. You've probably seen these at pet stores by now. They're all over the place because they're super cool. They're kind of expensive, but they're neat. But what they did was they added fluorescent genes into the fish. So you can see here, these are zebra. They're kind of normally a dull gray-brown color. And tetras can come in a lot of colors too, and they've even added into barbs, and they have all sorts of other ones now too that they're working on. But this was just step one of a bigger project. What they really wanted to do is first see if they can make them glow, or then glow in different colors. Because that's kind of the first step. Can we really do it? Will it actually work? So they went ahead and they did this, and they were like, oh, these look super cool. I would love to have these at home. So they actually went ahead and went through a process where the United States have now allowed them to actually sell these fish. The ones you would get at home can't have babies. They're infertile. Because that was one of the requirements that was necessary. Personally, I don't view it as a big deal. If these were to get out into the wild, breeding with the natural population wouldn't be very useful. Because normally, fish kind of want to have, gray, brown, dull colors so that they can hide in their environment. Whereas these are super bright and would never be able to hide in a natural environment. So I'm not too concerned about that issue as a lot of people seem to be. It's kind of weird to me, especially because most predators can see in the UV spectrum, which means these things literally glow to the predators. So things like a lot of predatory birds, they can see these things from a mile away. Literally from a mile away. So if these things even really got out, I don't think they would ever cause an ecological meltdown, because they would never survive long enough to breed, and even if they did breed, the offspring would never survive all that well. So they would never have an advantage over others, over the natural occurring versions. But yeah, so needless to say, they're infertile, but they do glow. You can put them under UV light and they'll glow in the dark, or you can put them under blue LEDs. Those blue LEDs will give them a really bright coloration. They're not, so to speak, glow in the dark, but they will glow a little bit. They'll fluoresce slightly. But the UV light's the best way to go, but you don't want to, leave them under UV light all the time. I've actually seen a lot of people do that, and it ends up killing the fish. A lot of them can get cancer if they even survive that long, so don't just UV light them all the time. Use the blue LEDs, and at night, if you're showing them off and want to be cool, then use the UV light for a while. But like I said, this was the first step in a bigger project. They just wanted to first see if they could work and make it glow, and then they were like, well, this is really cool, let's go ahead and sell these and make some money off the idea to help fund the research that they want to do. Now this is based on their concept of what they want to do. This is actually something I worked on in college for a course that was super cool called synthetic biology. It's a really neat new program kind of idea that's coming about recently, where it's kind of making your own organism. And that's basically what they did was synthetic biology. They created their own organism for their own purpose. But what we looked into doing, which was very similar to what they want to do, is to create a biological indicator. So there are stuff out there that do things, like I believe the one type of fish is called a rummy nose, which has a very bright red nose when it's healthy, and if the water conditions are good. But if the water's not doing great, the pH is off, the temperature's off, there's toxins in there, that red coloration disappears, and that's a really easy biological indicator, which you can buy those fish, believe it or not, at your pet store and actually use them. They're really useful because you can tell when your water's getting bad. But they wanted this on a bigger scale, something more impressive. So the idea, at least in this case, we wanted to detect lead in a water source. So we were trying to put this into algae, just because it's a lot easier to work with than fish are, and I don't know a lot about fish genetics, but algae, in particular blue-green algae, is not too bad. So the idea is that we would add in to a plasmid that we could put into that algae. We would add a lead promoter, which would be activated when lead comes in contact through a cascade, which would then cause translation to occur, which would make eventually the RFP, so red fluorescent protein gene. So the idea here is lead will attach to the cell membrane on a particular ion receptor for lead, which will bring that inside, attached to a lead binding protein. That lead binding protein leads a cascade that will activate the promoter, which will then begin transcription, which allows for that lead to be pumped out of an efflux protein. And efflux protein is just kind of, it removes certain things like lead from the inside of the cell and puts it out. But as a byproduct of this being translated, red fluorescent protein was added in. So normally you just have this lead promoter, the RBS, and then stop. We wanted to add in red fluorescence. So anytime lead is in the water, it activates these genes and red fluorescence occurs. So the cells will glow red. So if there's lead in the water, it is red under, a UV light. And this concept can be used for a lot of things, using what cells already naturally do. So like this lead efflux protein system, that's something that naturally can occur, and we just put it into a cell. Other cells have ways that they handle things like pH, where they'll use proton pumps and stuff like that. But all you have to do is have a promoter for whatever you're looking for, and attach it to a fluorescent protein. So in theory, if we had all these fish in the water, and we came over a pond and turned on the black light at night and saw that there was a bunch of red fish, we would know that there was lead in the water. Or maybe we attached a particular pH indicating system, so that if the pH was too high, they were blue. If the pH was too low, they're green. Or if there's mercury in the water, they're pink. Or, you could do all these different colors and make them color-coded for very particular things. So particular toxins. That was the idea of what we were working on here. In particular, we wanted to make a neat little, water test kit that you can attach inside your fish tank, and it would change colors under UV light based on what was going wrong. So, in this case, lead was in your water. It could be something like too much chlorine, high pH, ammonia, whatever you could dream of. And that's their idea too, so that they could have these transgenic animals out in the wild, in ponds and streams and whatnot. And scientists, instead of having to use a kit, just have to come by at night, turn on a UV light, and go, oh, look, the water is purple, or the fish are purple in the water. That means that there is, gasoline spilling in, or something like that. I don't know. So that was the idea, and that was just kind of a neat little concept that I absolutely loved. I fell in love with these glowfish once I actually started doing some reading on them when they first started coming out, and it's really neat. And again, it all comes back to fluorescent proteins. So that was our first installment. So that's GFP, Green Fluorescent Protein, and that's our first Science Sunday. So if you want to see more of this, let me know. Hit that like button. Leave me a comment with new topics. So comments, anything you can think of that you want to know more about. It could be anything even controversial. Cancer, it could be antibiotic resistance problems, all sorts of stuff that you whatever you guys want. I want to help answer your questions. So let me know what you think. Give me some new ideas, new topics that I can do a little research on, create a little PowerPoint for us so we can talk about it for a few minutes. And I hope you guys enjoyed. If you did, hit that like button so that I know that you enjoyed it. Pretty please. Don't forget to subscribe so that every Sunday you'll get that update that says we're doing Science Sunday, here's what the week's topic is. And if you guys really want more science stuff, I'll add more science stuff. So let me know in the comments below. Don't forget to subscribe, hit that like button. Hope you guys have a wonderful day. And remember, stay in trouble. Bye-bye.